the browser. We've also looked at how we can visualize genomic regions, so how to look for coordinates in Ensemble and look for information on genes and transcripts. Yesterday, we looked at the comparative genomics analysis data that we have in Ensemble. And I know that we didn't have time for the exercises. I do hope that you had a chance to have a look at them after the workshop yesterday or today. But if before I continue, I just want to ask if there are any questions. I also want to say there were a few questions in the living document yesterday. So thank you for asking those. I have responded to all of them so far. If you do have any other questions, feel free to ask them away. But if you don't, then I think we're good to move on for our session today. So today we are going to look at genetic variation. So what variation data we have in Ensemble, and we're gonna try one of our most popular tools, which is the Ensemble Variant Effect Predictor, or VEP. And this allows you to find out the consequences of your own variant. And hopefully we'll have time to do some exercises so that you can try and give it a go yourself. All right. So let's begin with a brief introduction to genetic variation. Let's first talk about variation types. So we can group variants according to their size or the number of nucleotides that are affected. Variations can come in a small scale where only one or a few bases of a gene are involved. So here on my slide, we've got two sequences. The top sequence is, let's say the top sequence is the reference sequence and the bottom sequence is our own data. So small scale variation types can include small insertions and deletions. We also call these dips or indels. So the first example, oh, sorry, I meant to change the effects of this. So this first example here is an example of an insertion. So in our reference sequence, we don't have a nucleotide associated at this position, whereas in our own data, we have an insertion of the G allele. Then the second example is an example of a deletion. So in our reference sequence, we have the C allele, whereas in our own data, we have a deletion at this position. And then we also have what we call single nucleotide polymorphisms, or SNPs. And these are just changes to the nucleotide. So this final example here is an example of a SNP. In our reference sequence, we have the A allele associated at this position, whereas in our own data, we have the T allele at this position. Variations can also come in a larger scale. So for example, in the chromosomal structure, these we call as well structural variation. And the types of structural variation are copy number variants, or CNVs, and large deletions, duplications, insertions, and translocations. And underneath, we have just a few images of what these would look like in the chromosomal structure. In Ensemble plants, we have variation data for 20 different plant species. These are listed on my slide here. So if you have the slides open, feel free to go through them, but I will also show you how to find them on the browser. And there is a question in the chat. So the person is asking to paste the link for the living document. Yes, that is a good point. So let me paste the link for the training page and the living document. All right, here we go. So the first link is the page for our exercises, and the second link is the one for the living document. This is where you can ask questions and find some more information about the course. Okay, so back to our presentation. So next I want to talk about the ensemble variation process. So where does the variation data actually come from? In general, we have 4 steps in the variation data process. So first, we import the variation data from publicly available archives. Then, during the import, the data is also being checked for quality. Next, we import the data that pass the quality control and link them with other databases or any associated studies. And then finally, we analyze the data and process them to make them available in Ensemble and for our users to view them. So let's just look at each step in more detail. So in the first step, we import variation data from publicly available archives and sources. So the keyword here is public. We want to make sure that all of our users have the same access to the data. So we only import from public archives and resources. So what we do is we search variation data from a number of different databases. And on my slide, these are just a few of these different databases where we import data from. But again, this depends on the different species. So depending on the species, we might take variation data from certain archives. But in general, we import variants from the European Variation Archive, or EVA, and then from any other species-specific projects and collaboration. In the second step, we apply some quality control measures on the data that we imported. So here, what we do is we map the variant to a reference assembly. We compare the reported alleles with the reference genome. And then we also exclude any suspect variants. So any variants that contain ambiguity codes or non-nucleotide characters. And you can read more about the different quality control measures that we apply in the URL at the bottom of my slide. In the third step, we link the variation data that pass the quality control to other databases. So here, what we do is we add value to the data. And what does this mean? This means that we link the variant to any allele frequencies, to any traits, phenotypes, and any publication data. Then in the final step of our process, we apply our own analyses on the variation data that we imported and that passed our quality control. So here we predict the variant consequence, we predict the protein function, we calculate linkage disequilibrium and variant conservation across different species. But how do we actually predict the variant consequences? So what we do is we take each variant and we plot them along the genome. And depending on where the variant falls within the gene, we predict the functional consequence of the variant at that position. So on my slide, what you're seeing is a gene. Just like in the browser, we have these horizontal lines depicting intronic regions and these boxes depicting exonic regions, where the yellow boxes are coding exons and the empty boxes are non-coding exons. So for example, if the variant falls outside of the gene, so let's say the 5' or the 3' region, the variant is unlikely to affect the gene itself, but it may affect the regulation. So for example, mRNA transcription and transcription activity. If the variant falls within the gene, but not within a coding sequence, so let's say any intronic region, The variant is also unlikely to affect the gene itself, but it may affect the mRNA stability. So, for example, splicing or translation efficiency. And then if the variant was to fall within the coding region of the gene, so within these yellow boxes here, um, the variant can have an effect on the gene in two different ways. So they can have a synonymous or a non-synonymous effect. If the variant has a synonymous effect, it means that the protein sequence remains the same, and therefore um, there is no change in the amino acid, and it is unlikely to affect the function of the overall protein. If the variant is a non-synonymous variant, or we also call them missense variant, there is a change in the amino acid and therefore in the protein sequence, and therefore we predict that it is likely that the variant has a greater consequence to the gene itself and the function of the protein. So to summarize, in Ensemble, we plot each variant to the genome, and depending where the variant falls within the gene, we apply or we assign a consequence to the variant. I don't expect you to read this table here, but what I want to show you is that variant consequences have special terms, which we call sequence ontology or SO terms. And it's just a standardized way of describing a variant consequence, just like how we have a standardized way of describing a gene function with gene ontology. If you are interested in the different SO terms or sequence ontology terms that we use, you can find the table at the bottom. You can find a link to the table at the bottom of my slide here. If you're interested to see the full table. When we talk about non-synonymous or missense variants, we rank them by their pathogenicity. So there are different algorithms that rank the changes in the amino acid sequence based on how conserved the protein is, based on the chemical change in the protein, in the amino acid position, and by looking at the 3D structure of the protein and its domain. So for example, we calculate the sorting intolerant from tolerant or SIFT score, which predicts the variant effect depending on how well conserved the protein is. So the SIFT score ranks from 0 to 1, with a score closer to 0 indicating a deleterious effect, and a score closer to 1, a tolerated effect. But please note that these are just predictions and not facts. So it's always good practice to validate your predictions experimentally in the lab. And then finally, I want to talk about reference alleles in variation data. So if you remember from yesterday, I talked to you about how genome assemblies are made. But one thing I guess I didn't mention was that genome assemblies are not made of a single individual of a population, but rather multiple different individuals. So it's kind of like a jigsaw puzzle from multiple genomes. So when you take a sequence from a genome assembly, and if you look at a particular variant, so for example, T in the sequence, it doesn't necessarily mean that it's a representation of all of the individuals of the species. So this T allele is found within this particular individual, but it doesn't mean that all the other individuals also have this T allele at that position. So this individual might carry an allele that could cause susceptibility in a disease. And therefore, when we talk about reference alleles, it's important to remember that it's not always the major allele or the allele that is found most frequently in a population. Most of the time, the reference allele is the major allele, but it's not always the case, and it's something to keep in mind. There are ongoing work that to replace reference allele with the major allele, but this, of course, takes time, so it's just something to keep in mind when looking at variation data. All right, so we are going to move on to a demonstration, and we are going to look at the wheat gene ending in 800, and we're going to find out how you can view a list of the variants within the gene. We're also going to look at the region and visualize variants within the region. And then we're going to look at a specific variant and find out more information about that. So let me just copy those variants and paste them into the chat. Oh, I think I've just deleted them. Oh, sorry, let me just jump out of my presentation. They've gone. Oh, here they are. Okay. So I've copied the variants and I'm going to paste them into the chat in case you wanted to follow along. So we're going to start with the gene and visualize any variants within the gene. So first, let's copy the gene ID ending in 800 and switch to our browser. So we want to go to Ensemble Plants. So go to plants.ensemble.org. I'll also paste that into the chat. And we are on the home page here. So as mentioned, there are two different ways to search. You can use the site-wide search in the top right-hand corner, but for more efficient searching, you can use the one in the green box here. So this is the one I usually use. First, you need to select your species of interest. So we were looking at Triticum aestivum, which is wheat, and then we can enter our ID just underneath here. Next, we want to click Go. And there's only one gene found in Triticum aestivum with this ID. So this is the ID, we've got the species, and then the location. So let's first look at the gene information that we have for this gene. So at the top, we can see which species we're looking at. We're looking at Triticum aestivum. Underneath, we have these different data tabs. So if you want to visualize the region, you want to go to the location tab. If you want to find any gene information, you go to the gene tab. And for any transcript information, so for example, if the gene is protein coding and you want to look at protein information, you need to switch to the transcript tab. But we are interested in the gene for now. So let's stay in this tab here. And in the left-hand panel, we want to move to the section genetic variation. So here, there are three options. We can visualize, or we can look at all of the variants within this gene in a tabular format. We can also visualize the variants as an image, or we can look at structural variants. In this case, for this gene ending in 800, we don't actually have any structural variants available, so that's why this is grayed out. Let's look at a table of all of the variants. So I'm going to click on variant table. And if you scroll down, you'll get table, which is still loading right now. Right. So after a while, once it's loaded, you'll notice that this table is quite huge. So you can keep scrolling down to see the number of variants that are found in this gene alone. So this table is very useful because for each variant, so for each line, we have different information. We've got the position, so first the chromosome, then the coordinate. We've got the alleles, so it's always the reference allele followed by the alternative allele. Then we've got the class, so what type of variant this is. This is a single nucleotide polymorphism or SNP. We've got the source, so this is EMS-induced mutation. In this case, there's no information, but we also give you the evidence and whether there's a clinical significance, but this is more useful for human data. So in this case, this isn't available. We've got the consequence type. This is the sequence ontology, or SO term, the standardized vocabulary for describing variant consequences. And if the variant is a missense variant or a non-synonymous variant, we also give you the amino acid coordinates and the amino acid change, and also the pathogenicity score. So you'll notice that the headers are looking a bit funny on my screen here, but this is simply because I'm zoomed in right now. So if you zoom out of the browser, you'll be able to read the headers properly. And just like in any other table in Ensemble, whenever you see anything that's underlined like this, that means that you can hover over it with your mouse and to find a description for that. So if you have a variant in mind, if you have a variant ID and you want to find it in the table, you can search for it up here using the search box. And you can also filter the table by column name. So for example, if you're only interested in missense variants, you can filter the table by consequence. So click on consequences. You get this pop-up window and you'll see all of the consequences are currently turned on. But to just select a single consequence, you need to turn all of them off first and then find the consequence you are interested in. So as an example, we're just going to look at missense variants. And then don't forget to click on apply. And that will update the table. And if you look at the consequence type column, so the one I'm in it right now, you'll notice that in this column or in the table now, this has been filtered to only include missense variants. So this is one way to filter the table. You can also apply multiple filters, so you can even select another column that you want to filter, and apply that as well. Okay, so this is how you view all of the variants in a gene. So let's visualize variants in the location tab next. So I'm going to switch to the location tab. If you scroll to the top of the page, we are landing by default on the region in detail view, and this is where we want to stay as well. So here on the right-hand side, if you scroll to the bottom, this is what we call the region in detail view. And we have this variant track already turned on by default here. So these individual lines, so they're lines rather than boxes because it's a very small coordinate, it's a very small region in the genome that's being covered. These individual lines are individual variants found in the gene, so the gene ending in 800. You'll notice we have different types of colors. So these are just describing what type of variants these are, and you can find a legend here underneath. And this is completely interactive as well. So for example, let's say you want to look at, again, missense variants in the gene. So you can click on the individual yellow lines, so these are missense variants, to find out more information about the variants here. So the one I just clicked on is a SNP. This is the allele, so the G is the reference allele and A is the alternative allele, and the consequence is missense variant. And don't forget that you can always zoom in as well. So click and drag your mouse, and then you can jump to the region, and you can find. You'll notice that the region I just highlighted actually has 3 different missense variants found here. All right. So this is. Hi, is there a question? Okay, I think that was just an accident in that case. Let's move on. So this is how you visualize variants in the region. Sometimes you can also find more tracks. I'm not sure if there are more tracks available for wheat. So I'm going to open the configuration tab, the configuration menu, and look at the different tracks we have available. So if you go to variation, we can see here on the right that we've currently turned on all of the variants, so all the variants that we imported from the different resources. But you can also visualize resource-specific variants. So, for example, any variants from serials DB only, or variants from the Watkins exome capture variant capture database. So you can always click on the eye icon and find a description that way as well. Underneath, we have some more information. So these are, again, more variant sets. So variants from different projects that were identified in different projects. And then there is a question in the chat. So the question is, can I download the data as TSV or text? Yes, so you can download the table that we looked at earlier, and you can also download the data in here. So if you want to download the data in the location, go to export data in the far left. And there are 2 things you can download. So you can download the sequence, or you can download the annotation. So all of the features that you are seeing in here. And there, for the features, there are different formats that you can choose from. So these also include comma separated or the tab separated format if you need that. If you want to download from the table, so let's go back to the table that I showed you earlier. If you want to download the data from this table, which is still loading here, there are 2 options as well. You can download it as a table using this Excel icon here in the far right-hand side, right-hand corner, or you can also export using, again, this button here on the left. So I hope that answered your question. I don't mean to delete it, so let me just close this window. Okay, so let's look for a specific variant in wheat now. So to search for a specific variant, we can go back to the homepage and start our search from the beginning. So we're going to select Triticum massivum, enter our variant ID. So this is just an example variant that we have found for this demonstration. And then we can click go. So we can see there's one variant found in wheat. This is the ID we searched for, and the variant was imported from the serial DB. So let's click on the variant ID. And at the top of the page, you'll notice we now have a new tab open, which is what we call the variant tab. And the variant tab, as the name says, gives you variant-specific information. So if you wanted more detail about the variant, for example, any population frequencies, you can find that in the tab here. So by default, you're landing on the Explore this variant page, and that's what you're seeing on the right-hand side. We've got some summary information about the variant. This variant is a missense variant, and this is the allele. So the first allele is always the reference allele, followed by the alternative allele. The highest minor allele frequency. We've got the location. We've also got the variant and variant calling format. So that's what you call this format here. We also have HGVS names, so that you can expand this option. There are different ways of writing variants down, and these are 3 of these. Any synonyms, so any other names under which this variant is known. Any external links. So again, this variant was imported from Serial DB, so we provide you with the direct entry for the variant and the original source and any more information. So down here, we have different variant-specific pages, and these pages here are the exact same pages as they are here in the left-hand panel. So it doesn't matter which ones you click, they'll still send you to the same page. So if you wanted to find out which genes the variant is found in, so what genes it's overlapping with, you can go to Genes and Regulation. And on the right-hand side, we have a table that will tell you all of the different genes that the variant is found in. So sometimes genes can overlap. So that means that a variant can affect multiple genes. But in this case, it only overlaps one gene, and that is the gene ending in 800. So this is the gene that we were just looking at there. So for this gene, we have information about the variant consequence, so how the variant affects the gene. We've got the allele, the consequence type. So this is a missense variant, causes a change in the amino acid sequence. So that means that there is a high chance that the variant has a greater effect to the gene function. We've got the positions, we've got the amino acid change, and then the codon as well, and any SIFT scores. So we can see in this case, we have a SIFT score of 1. So that means that the variant has a tolerated effect. So we're predicting that the variant has a very low effect on the function of the gene. All right. So on the left-hand side still, let's switch to the genotype frequency. And in the genotype frequency, this is where you find population frequency information. So how often is the variant found in a specific population? In this case, this variant was studied in the axiom population. So the variant, the T variant or the T allele is found this many times in the population, and the C allele, which is the alternative allele up here, is found this many times. We've got the genotypes, and sometimes we also give you the individual genotypes, so the individuals of the population. So if you click on Show under genotypes, often what you get is a list of the different individuals studied in the population and also their genotypes. So this individual from the population has the TT genotype, and that's the population they belong to. So what you could do here is you could look at the individuals, for example, that have a TC genotype and find out more information about them that way. Okay. Let me just double check I've showed you everything. Yes, all I wanted to show you. So let's switch back to the presentation. So we have time for exercises now. Um, so if you open the training page and go to the variation tab, we have a few exercises there. Let me show you. Variation and then open the exercises. There's only 3 exercises here actually. So you're going to look for this gene in wheat, and we're asking you to find a few different types of information for this gene, just to allow yourself to explore or get to know the browser a bit more. So I'll give you 15 minutes for this exercise, and then we're going to come back together and continue on with the variant effect predictor. Are there any questions? Okay, so there's one question in the chat, and the question is, how can we identify important features of the variant? So that's a very good question, and I guess um, this really depends on your research question. So, for example, some people might be more interested in the consequence itself. So, how does it affect the gene? Some people might be interested in which genes the variant affects. So, then you would go to the genes and regulation page and find out information about that gene and what it does. Some may be more interested in the different studies that the variant was studied in. So when it's available, we also give you publication links to any publications where the variant was studied. In this case, this variant wasn't studied in any publication that is available, so this is grayed out. So it really depends on the research question in this case. Okay, so 15 minutes for the exercises, and then we'll come back. If you have any other questions, please feel free to add them to the chat or to the living document. And if you have any questions now, please ask them. Hi, everyone. So time is not up yet, but there is a question in the chat, and I think it's a bit easier for me to actually show you how to do this. So there's a question that's asking whether we have any other cultivars available in Ensemble. So yes, we do. So for, I believe it's for rice and wheat, we have different cultivars available. Let me show you for wheat. So if you go to the homepage and go back to go to the link view full list of all species, you can search for the species you're studying. So for example, Triticum aestivum. And in the list, we have the reference. So this is the IWGSD one, but we also have a number of different cultivars here, and these are listed underneath. Um, if you want to see a full list or what cultivars you have available for the species, you can also just click on the species name to go to the species information page, which I'm waiting for it to load. Okay, so this is the species information page for wheat. And under genome assembly, we've got a section, other cultivars. So we can see we have 17 different cultivars available for wheat, and you can look at the different cultivars in a table by clicking View List of Cultivars. And scrolling down, we have these individual cultivars available as well. I hope that makes sense. Okay, so you still have a little less than 10 minutes, so we'll come back together in about 10 minutes. All right, a few other questions in the chat. So one of the questions in the exercises is what gene is affected by this variant and what's the amino acid change? So let me walk you through how you can find this. And I'm not going to use this example. I'm going to use the example we used earlier so that you can try it out yourself. So let me just go back. The example earlier that I used is this variant. So what you need to do is first look for the variant. So using the search bar, and then you need to open, oops, you need to open the variant tab like this one here. And to find out which gene it affects, you need to go to the genes and regulation page here on the left-hand side. So click on genes and regulation, and scrolling down, you can find a table. And in this case, this variant, this is not the same variant as the exercise, this is a different variant. This variant is found in the wheat gene ending 800. And because this is a missense variant, we know that there will be an amino acid change. So in the AA column, so amino acid, we have the change here, So this is from K to R, and you might need to use an amino acid table to find out what the different single letter coding stand for. I don't remember it myself, but you can just Google it and find out that way. So this is the amino acid change, K to R. And then just from earlier, if you've missed how you can find different cultivars for a plant, let me just show you again on my screen. So what you want to do is you want to go to the home page and open up the full list of all species. So in the home page, click on the link on the left that says full list of all species. And in here, you can find all of the species that we have available. But if you have a specific species you're interested in, so for example, rice, we can search for the rice scientific name, so Oryza sativa. If you type it in, sativa, and this will filter your table to include any rice genomes only. And you'll notice for rice, we actually have a lot of different cultivars available. But if you want to find, or if you want to list these different cultivars in a single page, you can also go to the species information page. So for example, if I click the Japonica group, this is the species information page, and you want to scroll down to genome assembly. And here we can see for this species, we've got an additional 15 cultivars available. And you can view this list of cultivars to give you a table of the different cultivars here. So you can look at example in locations, for example, and find information there. You can also go to the species page for the specific cultivar and search for genes in the cultivar specifically. And underneath, you'll also find what type of data we have available. So for this cultivar, we've got gene annotation, comparative genomics, but in this case, for variation, there is no variation database available for this cultivar. I hope that this makes sense. So you have a couple more minutes for exercises, and we'll come back together afterwards. All right, so it is time to move on now. So it's almost 8 o'clock India Standard Time. I know there is one outstanding question about the variant effect predictor. So don't worry about this because we are going to look at that now. If you have any other questions from the variation exercises, please feel free to add them to the chat or the living document. I will come back to you on that. But let's move on for now. So we're going to talk about the variant effect predictor, or we also call this VEP. So VEP is a tool that you can use to annotate and predict the functional consequences of your own variants. So let's say you just received your sequencing data and you've called your variants from the data, and now you want to actually find out more information, such as the consequences of the variants, how do they affect any genes, and so on. So this is exactly what you can do with VEP. You can find out the effect of your SNPs, your insertions, deletions, CNVs, or any other structural variants. And you can also compare your variants against a reference variant in this case. And this is how you use VEP. So first, you need to input your data into one of these different formats. Um, I will show you where you can find descriptions of these formats during the demonstration, but these have to be in either variant coordinates. So this would be the ensemble variant default format. We also accept the VCF. So this is the variant calling format, which is a very standardized format, and most of your variants would come in this type. We also accept HGVS, variant IDs, and speedy notation. So once your data is in one of those formats, you can run them through VEP. And what VEP does is it searches Ensemble and any other external databases to give you information about the effect of the variant. It gives you any pathogenicity scores if it's a missense variant, any frequency data, so how often is the variant found in certain populations, which genes, transcripts, or regulatory elements are affected. And if available, we also give you links to any publications for the variant. There are 3 different interfaces for VEP. You can use the web interface, which we are going to use today. So this is especially useful if you are just looking at a small number of variants, let's say 200, and you just want to have a quick look and see what the effect is. But we also have VEP available for the as a command line version. So this is very useful if you have large amounts of data and you want to run it on your own machine, or you can also access VEP via the REST API. So if you want to, for example, integrate the tool into your own pipeline. So with that in mind, let's move straight on to the demonstration. So we have 3 variants in wheat, chromosome 4b. These are the 3 variants. And we are going to use VEP to find out if the variants have already been annotated. So do they already exist in public archives? We're also going to look at what genes the variants affect and whether any of the variants affect regulatory elements. So the first thing you'll notice with our 3 variants is that these aren't really in a standardized format at all. So the first thing you will need to do is to put it into one of the formats that are accepted. And how can you find examples of these formats? So I'll show you here. You want to go to the Ensemble browser, and at the top of any page, there is a link to tools. So in the navigation bar, you want to click on tools. And then you want to go to the variant effect predictor. Oh. So let's go back and just try this link again. There we go. So whenever you see, this happened yesterday as well, but whenever you see the kind of 500 error, usually if you just refresh a couple times, it's back again. That just means that there's some connection issues with our servers. So once you click the variant effect predictor, you will be sent to the input form. But before we start adding our variants in here, I just want to show you how you can find examples of the different input formats that you can find. So click on variant effect predictor. So whenever you see this question mark, that means that you can open the documentation page for that data. So this gives you a more detailed description of how to use it, how the data was generated, and so on. But in this page, you can also find a link that says input format. And here are some examples of how these different formats look like, in case you needed a reminder. So we've got the default web input. So if you click on the link, that will just send you straight to that section with more description of how they look like. So default web input looks like this. And it's a tab separated file, and it's made up of columns and rows. So it's a table. And each row is one variant. So this is one variant, this is the second variant, and so on. And for each variant, we have information about the chromosome in the first column, the start coordinate, the end coordinate, the allele. So it's always the reference allele / alternative allele. We've got the strand. So if it's on the forward strand, it's plus. If it's on the reverse strand, it's minus. If you don't give any strand information at all, we just assume that your variant is on the forward strand. And then in the final column, we also have an identifier. So this is just an ID that you can give. So this is the VET default format. If you scroll down, we also have an example of the VCF. So this is what most of your variants would come in. So very similar to the default format, we've got the chromosome in the first column. Then we've got the position, the ID. So ID is always, it's always optional. So if you see a dot like this here, that just means that we haven't given it an ID. Then we've got the reference allele, the alternative allele, and often you'd have some optional information as well from your variant calling. All right, so let's, and if you scroll down, we also have HGVS. So HGVS, variant identifiers, and then speedy notation. So these are the different formats that you can choose for variant. Now, from our example in the slide, we have 3 variants, and we didn't have them in the right format. So what we did is we put them in the right format for you, and I will paste that into the chat. And in the chat, you'll see that this is the VCF, the variant calling format. So we have the chromosome, the position, we've given it an ID, the reference allele, and then followed by the alternative allele. So using the formatted variants, we can go back to our variant effect predictor tool and enter our input and select the different information that we want to get from Ensemble. So our variants are from a wheat example. So the first thing, once you're in the web tool, the first thing you want to do is to select your species of interest. So at the moment, I've got rice selected here, but we need to change this to wheat. So I'm going to delete the right species and click on Change Species. And then you can either enter your species name in the text box or start looking for your species in the list underneath. We have a wheat example, so we're going to search for Triticum aestivum. We're just going to look at the reference, sorry, but you can also select any cultivars if you're interested in them. And then you can click on the check icon to close the window, and then make sure in your species you indeed have Triticum aestivum selected. Next, we need to give our web job a name. It says it's optional, but I always recommend that you always give your jobs a name, because in, let's say, 3 months' time, you go back to your jobs, you look at them, you're not going to remember what you were looking at. So if you give it a good name, a descriptive name, it means that it will remind you what you actually did here. So I'm just going to call it wheat variance exercise. Next, in the input data section, you need to enter your variants of interest. So there are different ways to do that. You can enter them into the text box. So we can actually do this because we only have 3 variants, but you can also upload a file. So if you hover over this, you'll see that you can only upload files of up to 50 MB in size. If it's any larger than 50 MB, you will need to provide a URL instead. So often your institute would have an FTP site where you can upload your data to and provide a URL that way. But we have 3 variants, so I've just pasted them up here. Next, we need to, or we don't need to, but we can look at any additional configurations. So in these different sections, you can tell VEP what information you want to get back from the tool. So under identifiers, we've already got gene and transcript selected. So we want to see, or we want VEP to tell us what genes and what transcripts are affected. We can also look at proteins, if there are any UniProt features that are affected, and also give the HGVS notation. HGVS is a notation that is used quite often as well. So I'm going to turn that on. Next, we can open the variance and frequency data. So here, we can tell VEP if it should tell us whether the variant already exists or not. So by default, we always tell you if the variant already exists in the public database. And if you want to turn that off, you can do so by clicking on No. You can also give any variant synonyms. So just like genes, variants can have multiple different names. So if you turn this on, we will give you all of the known names that the variant is known as. Under additional annotations, you can tell VEP to give you, for example, information about the exon, intron, any protein matches, and so on. We don't have any regulation data available. So if it's empty like this, that means that there's no regulation data. So that in that case, from our exercise, we're not going to be able to find any variants that affect gene regulation. Then we've got predictions. So this is where you can retrieve any statistical information. So if the variant is a missense variant, you can turn on the SIFT score. We also have conservation calculations. And if you want to give information about any traits, so is the variant known to be associated with a trait, you can turn that on as well. And then finally, there's also a filtering option. So you can apply a filter before you even look at your results. But we always recommend not to apply filters until after you've looked at your results, just in case you might miss out on some interesting variants. Okay, so we've selected our species, we've named our job, we have our input, and we've selected our configurations. So next you can click on run, and that will send the job to our servers and is being queued to be run by our servers. So you see the status of your job. So currently it's queued. Every 10 seconds, this page gets refreshed automatically. So you can see that here in the name, and then it gets refreshed automatically. So once the variant has, once the job has started, this status will change and you'll know when it's finished. So right now it's running, and then the next status, it should be done. So it shouldn't take too long because it's only a very small job. There we go. So now you know it's done and it was successful. But before we look at results, you can let's just focus on the different options that we can apply here. So I mentioned on the first day, I think it was, that any jobs that you do in Ensemble only gets saved for 10 days. So after 10 days, your jobs get deleted from our servers, and then you won't be able to find them again. But if you have an account with Ensemble, you can save it to your account, and then that will be saved forever. So indefinitely, it's not going to get deleted. So if you want to save your job, make sure that you save it into your account by clicking this floppy disk here. And you can also share your web job with your collaborators by creating a URL. And then you can also delete the ticket. So there is a question in the chat, and they're asking about the privacy of the data that they post to web. So when you use web for sensitive data, for example, we always recommend that you use the command line version instead, because then you can be sure that your own server has the privacy settings that you need. So if you want to use web for sensitive data, please use the command line version and run it locally in your machine, because what's the word? We cannot be sure about the privacy of the data that's being saved in our server. So I hope that makes sense. Okay, so let's look at the results for our job here. So you can click on View Results. And we've got our results table, results page. And at the top, we have some summary statistics. So we can see here in this table that we ran 3 variants. 0 variants were filtered out. We didn't apply any filters. We can see that there is 1 novel variant. So that means that 1 variant has not been found in any public archive, and the other 2 have been, so they are known variants. All of our variants overlap only 1 gene, of which is 1 transcript. On the right-hand side, we have a pie chart of the different sequence ontology terms, sorry. So a third of our variants are missense, a third are 5' UTR, and then the other third, or the final third, are intron variants. All of the coding variants, so all of the variants that are found in coding exons, are missense variants. If you scroll down to the bottom, you will find your results in a tabular format. So the first thing I like to do when looking at results, I always sort my table by ID. So if you want to sort your table, you can just click on the column name. And now we have it sorted 1 through 3. Sorry, 1 through 3. And for each variant, we've got several information available to us. So the first variant that we looked at, this is the location, the coordinates for the variant. The allele is T, the consequence missense variant. This is the gene that it is affecting. Next, we've got these two columns. So the transcript that it's affecting is this ID ending in 200. What type of transcript this is? This is a protein coding transcript. We've got the exon numbers, so which exon the variant is found within. So 11, number 11, exon number 11 out of 18. We've got the HGBS notation. So this is what I turned on in my input form. Any cDNA protein amino acid positions, the amino acid change, codon change, any existing variants. So has this variant been identified in a public archive? And if it has, you will have an entry in this column. So this variant has been identified, the next one not. So the second one is a novel variant. And then the third one has been identified. So this is a known variant. We've got the reference allele. So for all of the variants, the reference allele is actually a C. The uploaded allele, so the second allele is the alternative allele, so our variant. The feature strand, and also any SIFT scores. So in our results, only the very first variant is a missense variant. So that's why only the first variant has a SIFT score. Remember, pathogenicity scores are only available for missense variants. Okay, so just above the table, we have an option to show and hide columns. So if there's any other information that you were looking for and that's not showing, the header might be turned off. So you just need to turn it on in this option here. If it's quite a big output table, you can also filter it using the filters up here. It's not quite useful here in our case because we only have 3 variants, but I just want to show you how you can do that. So to filter, you need to select the header or the column that you want to filter. So let's say you want to filter by consequence or sequence ontology term. Then you need to select the Boolean. So whether you're looking for that or not, and so on, or is it larger than, smaller than, equal to, and so on. Then you need to enter your term. Well, in our case, a term, but if you're looking for a number, you need to enter a number. So let's filter our table by missense variants only. And then you click add. Don't forget to add, otherwise it won't refresh the page. And once you refresh it, once the page refreshes, in your table, you should only find the filter that you applied. So we should only expect missense variants in our table. And indeed, you can see only the first variant is available in our results. You can download your results in different formats. So you can download it in the VCF format, in the VCF. You can also download it as a VET format, so this is the default format, or just as a text format. So this would be, I believe, a comma-separated file. There is also a question in the chat whether the tools are freely available. So yes, Ensemble is completely open source. It's freely available. You can use it in your research as you wish. Just remember to make sure to cite the most recent Ensemble paper for that. And if you are interested in the command line version of that, let me show you how you can find that. So what you want to do is go to the top of any page in Ensemble Plans, click on Help and Docs. So I need to click on More and then Help and Docs. On the left-hand side, we've got, find it first. Here we go. On the left-hand side in the panel, you want to go to genomic variation. And then, oh, it's not here. Hang on. Let me just find it. Ah, there we go. Sorry. It's not under genomic variation. It's actually under API and software. So under API and software, there is a link for variant effect predictor. And there here you can find the three different interfaces that we have available. I'll also paste this link into the chat because it's a bit difficult to find. I can just notice so that you have an easy access to it. Okay, we do have a couple more minutes, but I am finished with my demonstration. It's unfortunately not enough time for exercises, but if you have any other questions, please feel free to ask them away. If you don't have any questions, please have a go at the variant effect predictor. Try to maybe just run a job, just one job in the exercise, and become familiar with the tool. If you have any questions in the meantime, so after the workshop, for example, you can enter the question into the living document, and I'll answer them there. Or you can ask them tomorrow, first thing tomorrow morning. Sorry, morning. First thing tomorrow in the workshop. Or if you want me to go through anything, you can ask me to demonstrate it again for you, and so on. But for now, I'm finished with the demonstration. Are there any questions, any outstanding questions? Oh, there was one question that I missed there, and the person couldn't find VEP in Ensemble Plans. So let's say you are in the home page. What you want to do is go to the top of the page in the navigation bar, click on Tools, and in Tools, it's the first one in the list. So Variant Effect Predictor. And this is how you get to that job. I already have one lined up here, but you can just start a new job and enter your input in here. Let me also share that link with you in the chat. Hey, any other questions? All right. If there are no other questions, I don't think there are any outstanding questions either. I'm happy to finish our session a bit early today. Please do try the exercises, and if you have questions for tomorrow, we'll have some time before we look at the Biomart tool. So just as a reminder, let me show you the agenda. So tomorrow, it's going to be quite a short session, actually. We're going to be joined by Ian, my colleague. He's going to talk you through how you can use Biomart. So Biomart is another tool of ours, and what you can do with it is you can find information about a list of genes or coordinates or proteins to download in bulk without needing to know any programming language. So often in genomics, if you want to find information about from a big list of genes, you will need some kind of scripting knowledge, but Biomart gets rid of this. So Biomart has a user interface, and you can do bulk queries without needing to know any scripting at all. We're also going to have some time for feedback. So I will add, if you want to retrieve an attendance certificate, you can do so by filling out a feedback, and I will give you time for that tomorrow. So don't worry about it for now.
Okay. So, it's all over for today from your side.
Yes,
Yes.
I'm
Thanks,
finished for
ma'am.
today. Thank you.
Thanks for your efforts for delivering in this workshop. And we will meet again tomorrow at the same time, 7 p.m. for the final session by ensemble team. So thank you, ma'am. Now I seek your permission for ending today's session.
That's perfect. Go
Thank you.
ahead. Thank you, everyone.